we will go to the processes which are involved in the gastrulation in frog embryo. So, gastrulation is a process in which a single layered blastula is transformed into a double or a triple layered gastrula. So, first we will go to the structure of the blastula before we go to the gastrula. So, this blastula, it is a ball of cells. It is a hollow ball of cells which is having a cavity inside that is called as the blastocele. Now, we will just have a look at the fate of the blastula. That is what is called as the fate map. So, fate map is a chart which shows the fate of each part of an early embryo, that is the blastula. So, this was developed by Walter Vogt in 1925, and this can be traced through artificial staining techniques. So, accordingly, there are 3 prospective centers in a frog blastula. That means there are 3 prospective areas which give rise to certain parts of a future embryo. So, these are called as the prospective centers. They are the prospective ectoderm. So, this is the prospective ectoderm. Here, this is the prospective ectoderm. So, this is the dorsal view and this is the lateral view where we can see the prospective ectoderm, the prospective notochord and mesoderm. So, here you can see the notochord and the mesoderm and the prospective endoderm, because amphibians are triploblastic organisms. So, the gastrula will have 3 germ layers, that is the ectoderm, the endoderm and the mesoderm. So, this part is the prospective ectoderm. This part is the prospective endoderm, and this is the prospective notochord and this is the prospective mesoderm. So, these are the various areas which you can see in the fate map of the blastula. So, first we go to the prospective ectoderm. The prospective ectoderm is this area which is a pigmented area seen on and around the animal hemisphere, because animal hemisphere is more pigmented compared to the yolky white vegetal hemisphere. So, this area is called as the ectoderm. This consists of 2 regions. One is called as the neurectoderm. The other is called as the epidermal ectoderm. So, here in this diagram you can see the neurectoderm and the epidermal ectoderm. The neurectoderm, it is that prospective area that develops to the central nervous system of the future embryo. So, this develops to the central nervous system of the future embryo, which has got a region that develops into the sensitive layer of the eye also. Now, the epidermal ectoderm. The epidermal ectoderm, or this area, it gives rise to the epidermis of the skin, and there are some sub areas in this epidermal ectoderm which will give rise to nose, ears, suckers of the tadpole and the stomodium, or the area surrounding the mouth. So, it is from the epidermal ectoderm that areas such as the nose, ears, suckers and stomodium develop. So, during gastrulation, which we are going to study now, the epidermal ectoderm, it will spread downwards and converges towards the vegetal pole. So, it will spread downwards and it converges towards the vegetal pole, and that is why the epidermal ectoderm is called as the zone of expansion. So, we will see that movement of the ectoderm during gastrulation, and this was described by Gilchrist in 1968. So, that is regarding the prospective ectoderm area, which consists of the epidermal ectoderm and the neural ectoderm. Next is the prospective notochord and mesodermal areas. So, this is represented by a crescentic area. So, this is the crescentic area of grey color below the prospective ectoderm. So, it is seen below the ectoderm. So, this crescentic area, this much crescentic area represents the prospective notochord and the mesoderm. So, first we go to the notochordal area. So, this is the notochordal area. So, here in the lateral view you can see the notochordal area. the notochordal cells, they occupy a large portion of the dorsal side of the grey crescent. Grey crescent area is that area which is opposite to the sperm entry because of the movement of the cortical granules away from there. So, this is the dorsal side of the grey crescent where the notochord cells occupy a large area, and just beneath the notochord towards the vegetal pole. So, just beneath the notochord towards the vegetal pole, a narrow strip of cells can be seen. It is called as the precaudal plate. So, this area, this prospective notochord will develop into the notochord of the embryo. The precaudal plate is that which gives rise to some parts of the head mesoderm. So, the mesodermal tissues of the head is arising from the cells of this precaudal plate. So, here you can see the notochord in the dorsal view and the precaudal plate. The notochordal area gives rise to the future notochord, and the precaudal plate gives rise to some of the head mesoderm. Now, coming to the mesoderm. This is the prospective mesoderm or the future mesoderm. Here you can see the future mesoderm. It is seen on either side of the notochordal area. So, this is the notochordal area. So, on either side you can see the future mesoderm. So, as you can see from the dorsal view and the lateral view. Now, the prospective endoderm. The prospective endoderm is this area which is seen in and around the vegetal pole. So, this is the vegetal pole. So, this area represents the endodermal area, the prospective or the future endodermal area. Cells in this area will later develop into the midgut and the hindgut of the embryo. So, the prospective endoderm develops into the midgut and the hindgut of the embryo. The endodermal cells which are just below the precaudal plate area, that is here, the endodermal cells at the mid dorsal point of the grey crescent, they give rise to the endodermal lining of the mouth, gill region, pharynx, etc. So, this region, this part of the endoderm will give rise to the endodermal lining of mouth, gill region, pharynx, etc. So, this is called as the pharyngeal endoderm. This is the pharyngeal endoderm, whereas the other parts they give rise to the midgut and the hindgut. So, here this is the pharyngeal endoderm. This part is the pharyngeal endoderm. So, this is a fate map of the frog Blastula. Now, we will move on to the process, that is gastrulation. So, gastrulation is a process in which a single layered blastula is transformed into a double or a triple layered gastrula. And how this is made possible? This involves movements of cells in various directions, changes in the physical tension of the cells, changing their shape and structure, and also alterations in their metabolic processes. So, the gastrulation process is brought about by the movements of cells, primarily by movement of cells, then by changes in the physical tension of cells, their turgidity, etc., and alterations in their metabolic processes. So, what do the cells do? The cells will move actively from one place to another to take definite places in the embryo. Earlier, it was not an organized structure. The blastula was not an organized structure, but once gastrulation starts, the cells move in different directions in an organized way and occupy certain positions in the gastrula, which will develop into future regions of the embryo. So, now it is a differentiated condition. That is what is meant by gastrulation, and this is made possible with the help of cell movements. So, because of cell movements, what happens? There will be formation of definite cell layers by the mass migration and the rearrangement of cells. Earlier, the blastocele was only a solid ball of, the blastula was only a solid ball of cells with a central cavity called as the blastocele. The cells didn't have their future fates recorded on them, but once gastrulation starts, their fates will be determined. So, such movement of cells to develop into prospective areas of the embryo to form a an organized structure. Those movements are called as morphogenetic movements or formative movements. Why it is called a morphogenetic movement? Because such movement of cells will determine the future structure of the embryo. So, these movements determine the development of the future structure of the embryo. That is why it is called as a morphogenetic movement. Now, there are three types of movements which are involved in the gastrulation in frog. First one is called as invagination. Second one is called as involution. Third one is called as epiboly. So, first is invagination, second one is involution, and third one is epiboly. Invagination means pushing in of cells. An area of cells will get pushed in. That is what is called as invagination. Involution is the rolling movement of cells to the interior. When cells roll over into the interior, it is called as involution. Epiboly is the spreading of cells over the surface of the embryo. So, the movements are invagination, which involves a pushing in of cells altogether. Involution is the rolling in of cells to the interior, and third one is epiboly, which is the movement of cells or the spreading of cells over the surface of the embryo. So, the first movement is called as invagination. Invagination is shown by the endoderm. So, as you can see, this is the blastula, this is the blastocele. So, we will go to an early to a blastula which is ready for invagination. So, this is the blastula, this is the vegetal pole, this is the animal pole, and when you come to the fate map, it is this region which represents the endoderm. This region represents the ventral side, that is the vegetal pole represents the endoderm. So, we will go to the process first, that is during invagination. So, this is the vegetal pole where there will be the endoderm, the future endoderm, as we observed from the fate map. So, here was the ectoderm towards this region, towards the grey crescent area was the notochordal area, prospective notochord, and the prospective mesoderm on this side, and this region was occupied by the endoderm, future endoderm. So, this is the vegetal pole. The cells of the vegetal pole form the future endoderm as it was mentioned in the fate map. So, the first process is the formation of a small notch. So, this is a small notch which develops below the equator. This is the equatorial region, this is the equator. It develops below the equator, exactly below the mid dorsal point of the grey crescent of the blastula. So, it is this region that represents the grey crescent of the blastula. So, it is this region that represents the grey crescent of the blastula. Just below that, a notch appears. So, the notch appears below the equator, exactly below the mid dorsal point of the grey crescent of the blastula. So, this is the mid grey crescent area. Cells of this area will move inward towards the interior, and this notch that develops is called as the blastopore. So, the notch is called as blastopore. So, a small notch develops, the cells of this area move inward to the interior, or there is a pushing in, and this notch represents the blastopore. Now, what happens? This invaginating layer, it develops a space inside. So, you can see a space is developed inside this invaginating layer. This space is called as the Archenteron or gastrocele, which opens out by the blastopore. So, the Archenteron or the blastocele or the gastrocele, not blastocele, the gastrocele or the Archenteron, it opens out through the blastopore. So, this is only an initial stage of invagination, where you can see the blastocele here. So, by this invagination, a new cavity or the Archenteron or the gastrocele develops. Now we will go further. So we will see what happens further. So here again we come to another diagrammatic representation where you can see this is the blastopore. So this is the presumptive or the future endodermal area where the blastopore or the notch appears and the cells start moving in. Now here you can see this invagination is growing and this cavity is also growing, and this cavity is called as the Archenteron. So this Archenteron, which is initially slit-like, later will become crescentic or horseshoe shaped and finally become circular. So you can see the shape, change in shape of the Archenteron. Initially it is only a slit, initially it is only a small notch, then it becomes slit-like and finally it becomes circular. So the invagination is extending circumlaterally to the sides also. Now this blastopore is having lips. So this is the invaginating. So this is a small invagination. Now it will develop lips. This lip is called as the dorsal lip. So this is the dorsal lip of the blastopore. This is the ventral lip of the blastopore, and though you cannot see them in this diagram, on either side will be the lateral lips. So they will be on the either side. So this is the dorsal lip, the ventral lip, and there will be lateral lips for this opening. The blastopore possesses 4 lips: dorsal, ventral and lateral lips. Now what happens? So endoderm is filled with yolky material, and it is this endoderm that is invaginated. And these yolky endodermal cells, so this is all yolky endoderm which is seen towards the grey crescent area. They become separated. So what happens? They become separated from the outer layer. So here you can see a small bridge like area, the endoderm moving up, or it is forced upward. And what happens to the blastocele? Initially this is the blastocele. This is the position of the blastocele where you can see in the second diagram also. But once these yolky cells, they are pushed upwards. When the yolky cells are pushed upwards, the blastocele will be displaced to this direction. Earlier blastocele was occupying this position, but because of the pushing up of these yolky endodermal cells to the upper region, the blastocele becomes displaced to this direction. That means the vegetal hemisphere cells are undergoing a pushing in. So this is called as invagination. So the endodermal area lying in the marginal zone is taken to the archenteric wall by the process which is called as invagination. Now we will go to the second process, that is involution. So what is involution? Involution is the rolling in of cells. So for that we will come to the diagram again, where this is the epidermal ectoderm, this is the neural ectoderm, the prospective neural ectoderm, this is the notochordal area, the prospective notochordal area, this is the precaudal plate, and this is the blastopore. So first what happens is the pharyngeal endoderm, that is just above the, this is the pharyngeal endoderm which is seen just above the blastopore. It will roll over the dorsal lip of the blastopore. For that we will again have a look at the pharyngeal endoderm. So I told you this is the notochord, this is the precaudal plate, this is the endoderm, where this region is the pharyngeal endoderm. So it is this, and this is the blastopore. As you can see, this is the blastopore in dorsal view. So this blastopore is formed by the invagination of the endoderm, but this area, that is the prospective pharyngeal endoderm, will roll in through the blastopore. That is what was earlier mentioned. Coming back to involution again, the prospective pharyngeal endoderm. So this is the prospective pharyngeal endoderm. That means the area just above the blastopore, as we saw from the fate map. So this is the prospective pharyngeal endoderm. It is this pharyngeal endoderm that first involutes over the dorsal lip of the blastopore. So this is the dorsal lip of the blastopore. So you can see in this diagram that it has reached here. So it is by the rolling in of cells. Initially it was in this position, that is, it was near the dorsal lip of the blastopore, but now it has rolled to the interior through the dorsal lip of blastopore and has reached this position. That is what is called as involution. So it is occurring by the rolling in of cells. Next is the rolling in of the next area. So this is the first area to roll in. So automatically this will be the second area to roll in, and that is the precaudal plate. So the precaudal plate will now roll in. Now this is followed by the prospective notochord, and the tail mesoderm is also there. The hind end of the mesoderm also, that will also roll in. So what all things undergo involution? First the pharyngeal endoderm, followed by the precaudal plate, which is followed by the notochord and the hind end of the mesoderm, that is the tail mesoderm. So we will just have a look at the tail mesoderm again from the fate map. So here, this is the mesoderm, and this can be considered as the tail end of the mesoderm. So first, this is the pharyngeal endoderm. Here in this diagram, this is the pharyngeal endoderm. So it is the pharyngeal endoderm that rolls in, then the precaudal plate, then the notochord and the tail mesoderm. All these things, they roll inward. So once again, the areas which undergo rolling in are first the pharyngeal endoderm, then here the pharyngeal endoderm, the precaudal plate, the notochordal cells and the tail mesoderm. They all roll in. So as these materials move inward around the dorsal lip, they become stretched and elongated greatly, because here in this diagram, this is the pharyngeal endoderm, this is the precaudal plate, this is the notochord which has started rolling in, but here you can see they have elongated, they have stretched and got elongated greatly. So now here is the pharyngeal endoderm, here is the precaudal plate, and here is the notochord and some area of the mesoderm. Now what happens to the Archenteron? The Archenteron has now increased its area. It has now become somewhat circular. So this pharyngeal endoderm which is first involuted, this is the pharyngeal endoderm which is first involuted, it forms the anterior most part of the archenteric wall. So this is the wall of the Archenteron, whose anterior most part will be occupied by the pharyngeal endoderm which has first involuted, just first rolled in. Then the precaudal plate becomes a part of the archenteric roof. So this part is the precaudal plate that has become a part of the archenteric roof, and behind is the notochordal region. So the notochord also stretches anteriorly along the dorsal side of the Archenteron, and it actually forms a mid-dorsal strip of the archenteric roof. So it is forming the mid-dorsal part of the archenteric roof. So it is occupying a mid-dorsal position, this notochord. So the Archenteron is now having an anterior wall made of the pharyngeal endoderm, then the precaudal plate, then the notochord which is occupying the mid-dorsal roof of the Archenteron, and the other sides will be occupied by the endoderm. The mid-dorsal side will be occupied by the notochord, whereas all the other sides of the Archenteron will be occupied by the endoderm, because you have to imagine this is only a section. So the only this mid-dorsal region will be having the notochord, other areas of this Archenteron will be covered by the endoderm, of course. Then, so by this time the cells of the posterior part of the notochord also roll over the dorsal lips of the blastopore. The posterior side of the notochord also reaches this region, and they will form mid-dorsal strip of the archenteric roof. So, this is the anterior part of the notochord, this is the posterior part of the notochord which has just now rolled inward, and they form the mid-dorsal roof of the archenteron. So, you have to imagine a cavity, the archenteron. You have to imagine this archenteron as a cavity which is having a wall made of endoderm. The whole wall is made up of endoderm. You have to imagine a ball or a disc which is having a wall made of endoderm, but on the dorsal side there will be the strip of notochord. There will be the notochordal strip along with the precaudal plate. All the other region will be enclosed inside the endoderm. Now, after entering the embryo, the entire notochord as well as the mesodermal material, they move from the posterior end towards the anterior end as a single sheet. It is called as the chorda mesodermal mantle. So, the notochord as well as the mesoderm has moved in or rolled in, and they form a continuous, they form a single sheet which is called as the chorda mesodermal mantle, and this mantle will now penetrate between the ectoderm and the endoderm. This mantle will penetrate between the ectoderm and the endoderm. Now, so that we can see through a diagram, that is, here this is the mesoderm, this is the endoderm, and this is the ectoderm. So, now it has occupied a position in between the endoderm and the ectoderm. Before coming to this diagram, we will also go to the other movements which have to follow. So, the mesoderm is also rolling in. You can see here through the ventral lip of the blastopore, the mesoderm rolls in. Here you can see. So, when the notochordal material was moving along the dorsal lip of the blastopore to the interior, through the ventral lip of the blastopore, the mesoderm is also rolling in. So, involution is done by the notochord as well as the mesoderm. The notochord is involuting through the dorsal lip, whereas the mesoderm is involuting through the ventral lip, and it is slowly occupying the position between the endoderm and the ectoderm. Now, what all mesodermal tissues have rolled in? The trunk, the somatic, and the ventrolateral mesoderm. They are the ones which roll over the ventral lips of the blastopore. So, the trunk, the mesoderm which will form the trunk, the somatic tissues and the ventrolateral areas will roll over the ventral lip of the blastopore. So, the notochordal material, the precaudal plate, etc. have rolled in through the dorsal lip as well as the pharyngeal endoderm, and the mesoderm is rolling in through the ventral lip, and you can see slowly it is occupying the position between the ectoderm and the endoderm. So, they form the mesoderm as well as the notochord, they will form the chorda mesodermal mantle which is occupying the position between the ectoderm and the endoderm. So, the mesoderm, it will occupy the whole area between the ectoderm and endoderm except at a small portion of the anterior region of the embryo. So, there will be a small portion of the anterior region of the embryo where the mesoderm does not occupy the space between endoderm and ectoderm, and it is this mesoderm free area that will develop into the mouth of the embryo. So, that was the second process called as involution. So, first was invagination of the endoderm, which formed a cavity called as the gastrocele or the archenteron, and the notch which initiated invagination is the blastopore. It is the presumptive endoderm that underwent invagination. Along with that, there was shifting of the blastocele, there was displacement of the blastocele and reduction of the blastocele. Then along with that, there was involution of cells or the rolling in of cells. So, first it is the pharyngeal endoderm that has involuted, followed by the precaudal plate, the notochord. The mesoderm also undergoes involution once the endoderm has moved inwards. Once the endoderm is fully pushed inwards, the mesoderm starts involution and it starts occupying the place between the ectoderm and the endoderm. So this is the ectoderm, this is the endoderm, and you can see slowly mesoderm is trying to occupy the position between the ectoderm and the endoderm. And meanwhile the endoderm is also moving in, thereby reducing the blastocele to a small cavity, or you can see an obliterated blastocele. Now the third one. Third one is called as epibolic movement. Epiboly is the spreading of the presumptive ectoderm on the surface of the blastula. That means ectoderm has got two portions. One is the epidermal ectoderm and the other is the neural ectoderm. So what happens is the cells of the ectoderm, they will start moving over the surface of the blastula, and the ectoderm will slowly take up the position of the prospective endoderm and the cauda mesoderm that has moved inside. That means the endoderm has moved inside. The cauda mesoderm has also moved inside. That means the notochordal tissues, future notochordal tissues, the future mesodermal tissues have also rolled inside. Endoderm has also moved inside, and the exterior of this embryo will now be occupied by the ectoderm because of its spreading or because of the expansion of the ectoderm. So here initially you can see this part is occupied by the endoderm, this part is occupied by the notochord, this part is occupied by the precaudal plate, and here there is a small strip of mesoderm. But now once the invagination and involution processes are going on, the ectoderm will spread over the surface of the embryo, and finally you can see that the outer region is now occupied by the ectoderm, whereas the notochordal material, mesoderm, endoderm has all moved inside and the blastocele has obliterated. So the expansion of the ectoderm is considered to be an active process occurring at the expense of the thinning out of epidermal ectoderm in all directions, because here the ectoderm was thicker, but now it is thinning down by the movement of cells in all directions over the surface of the embryo. So that is what is called as epiboly. Earlier it was a thick layer which occupied only this portion of the embryo, but once the cells move in all directions along the surface of the embryo, it becomes thinner, but it comes to occupy the whole surface of the embryo. So this is called as epiboly. So epiboly is also a factor which will push the endoderm inside. The pushing of the endoderm to the interior is also facilitated by the epibolic movement of the ectoderm. Now, coming to the expansion of this area which is called as the neural ectoderm. The neural ectoderm, its expansion occurs only in the longitudinal direction, only towards the blastopore. So the neural system, the presumptive neuroectoderm is only expanding only in the longitudinal direction towards the blastopore. So only this area will be occupied by the neuroectoderm. All the other area will be occupied by the epidermal ectoderm. Only a small area, a longitudinal area on the dorsal surface will be occupied by the neuroectoderm, but the whole other region of the embryo, the whole surface of the embryo other than this neuroectoderm will be occupied by the epidermal ectoderm. As I already told you, the ectoderm is divided into the epidermal ectoderm and the neural ectoderm. The neural ectoderm will now occupy only a small, only a longitudinal area in the anterior posterior direction on the dorsal side, whereas all the other area will be occupied by the epidermal ectoderm. Now we will again have another look at the gastrulation process, the same process which you can see through other diagrams. That is, here this is the blastula with the vegetal hemisphere, the animal hemisphere, the blastocele. This is the grey crescent area of the blastula. Just below the grey crescent area, a small notch develops, and that is the blastopore. This is the dorsal lip of the blastopore, and this is the ventral lip of the blastopore. So this process that has started is called as invagination, which will create the blastopore. Then Next, you can see the expansion of this cavity. This is the Archenteron or the gastrocele, which is formed from the blastopore, which opens out through the blastopore. This is the yolk laden area. You can see the blastocele is getting reduced because of the expansion of the Archenteron. In the next diagram, you can see the blastocele is further reduced and displaced to this position because of the movement of the yolk, as it was already mentioned. Since the yolk moved upward, the blastocele is displaced to this direction. The Archenteron, it is expanding. Now you can also see the chorda mesoderm. It is involuting. Involution of the chorda mesoderm. This is the pharyngeal endoderm which has involuted first. This is the chorda mesoderm which has involuted. And you can see a much obliterated or reduced blastocele here. You can see an expanded Archenteron here. This is the dorsal lip of the blastopore through which the chorda, the notochordal tissues have involuted in. So this is the notochordal area. And this is the endodermal area, and the Archenteron is surrounded by the endoderm, and you can see the mesoderm is gradually involuting in through the ventral lip, occupying position between the ectoderm and the endoderm. This is endoderm, this is ectoderm, this is mesoderm, which is trying to occupy the position between the ectoderm and the endoderm. So far the processes we discussed were just repeated through these diagrams. Now we will go to the next stage, one of the important events which you can see in frog gastrulation, that is called as the yolk plug stage. What is meant by yolk plug stage? You can see, so this is the blastopore, this is the blastopore, or we will go to the previous diagram for a better understanding. So this is the blastopore, where this is its dorsal lip and this is the ventral lip. You can see that a plug-like mass of endodermal cells, that is the yolk-laden macromeres. The yolk-laden macromeres are actually formed in this area. You can see a plug-like region, a plug-like mass of endodermal cells which are yolk-laden macromeres. They are seen to be filling the rim of the blastopore. So just like we plug the mouth of a bottle with the help of some stopper, you can see a plug which is formed of yolk-laden endodermal cells occupying the rim of the blastopore. This is called as the yolk plug. This yolk plug is formed of two reasons. One, because of the spreading of this ectoderm. So the spreading of epidermal ectoderm all around the embryo will lead to a convergence of a small amount of this endoderm through the blastoporal rim. So this is one reason. The pressure caused by the spreading of the ectoderm, the epidermal ectoderm, causes some amount of yolk-laden endodermal cells to come out through this blastoporal lip, or to stick out or project out through the blastoporal lip, and this is called as the yolk plug. So one reason is the movement of the ectoderm. Second reason, the area of cells rolling inwards is increasing. First the pharyngeal endoderm rolled in, then the notochord rolled in. So the dorsal lip of the blastopore is also growing downwards. The dorsal lip of the blastopore is also growing downwards because of the involution. And now the lateral lips of the blastopore will also be expanding, and the lateral lips, so earlier the blastopore was just like a small opening, but because of the expansion of the lips, the dorsal lip and the lateral lip, what happens is that the blastopore will assume a ring-like shape. Earlier it was a small pore, but now the blastopore looks like a ring through which the yolk-laden macromeres will project out. So now there is more space for this yolk-laden macromeres to come out. That is why you can see them in the form of a plug. Earlier it was not possible because the blastopore was just like a slit. As you can see here, this is the blastopore which was just like a slit where the dorsal lip and the ventral lip were close together. But later, because of all these cell movements, the dorsal lip is here, the ventral lip is here. That means the blastopore has expanded. The radius of the blastopore has expanded, so the yolk plug comes out. So these are the two reasons. So one is because of the spreading of the ectoderm, and second one is the increase in the diameter of the blastopore. Earlier it was just like a slit, but now it has become like a ring. So the yolk-laden macromeres are seen protruded out. And not only that, they are too large to be incorporated immediately in the floor of the Archenteron. So there is not enough space for all of them to occupy the floor of the Archenteron. So they have to wait before they can occupy the floor of the Archenteron because they are larger cells. So for the time being, they will be seen protruding out, and that is what is called as yolk plug, and it is only a temporary phenomenon. That means it will close. Why is this yolk plug very prominent? Because all the other areas are pigmented, whereas the cells of the yolk plug are white colored because of the yolky content, whereas the other regions are pigmented. So it is a real contrast. The cells of the pigmented dorsal lip as well as the ventral lip are in sharp contrast with the yolk-laden white endodermal cells. That is why you can prominently see the yolk plug. Gradually, there will be space created in the inner side of the Archenteron, so the endodermal cells can move inward. The yolk plug is withdrawn because there is now more space created inside for these endodermal cells to lie beneath the Archenteron, because the endoderm has to form the floor of the Archenteron. So slowly the yolk plug will come in. It will be withdrawn to the interior, or it is retracting. The rim of the blastopore also contracts. Earlier the rim was larger, so the yolk plug could be seen outside, but now the rim is contracting, so the yolk plug slowly retracts to the interior. And finally the yolk plug completely disappears from view due to the closure of the blastopore, because the blastopore will finally close and the yolk plug will completely disappear from view. So yolk plug formation is only a temporary phenomenon, but it is a characteristic feature of frog embryogenesis. Now we have to mention what happens to the blastocele. So you can see this is the blastocele. Earlier this was the blastocele. So this was the blastocele and this was the gastrocele. But now, because of the expansion of the gastrocele or the Archenteron, and because of the pushing up of the endoderm, the blastocele has been displaced to this direction. And finally, when the Archenteron grows, what happens to the blastocele? It will be reduced. It becomes displaced to the anterior direction. This is the posterior region of the embryo and this is the anterior region of the embryo. So the blastocele has been displaced to the anterior region, and it is shifting in the anterior ventral region because of the expansion of the Archenteron. And now this blastocele gets reduced to a slit between the endoderm and the yolk-laden cells. So it is only a small slit now, and there is a layer of cells which will separate the Archenteron from the blastocele, and this layer of cells which separates the blastocele from the Archenteron is called as completion bridge. It is only a very small thin cell layer, and sometimes this cell layer will rupture, thereby the blastocele becomes part of Archenteron. So in some cases the blastocele will merge with the Archenteron by rupture of this bridging cell layer between these two. So these are the various events occurring during gastrulation, and the final gastrula will enclose a cavity, and this cavity is called as the Archenteron, which will become the future gut of the embryo. It will become the future gut of the embryo, and this has got two layers now, an outer layer and an inner layer. The outer layer consists of a mid-dorsal neuroectoderm and epidermal ectoderm around the embryo, As I told you, we will go to the diagram which we earlier saw, that is here. This is a final gastrula which has got two layers. The outer layer, this is the outer layer which consists of the new ectoderm only in the mid-dorsal region. So imagine this as a ball. You imagine the dorsal side of that ball in the longitudinal direction. Over the dorsal side of that ball there will be a strip of ectoderm, that is called as new ectoderm. All the other surface, all the remaining part of the surface of the embryo will be occupied by the epidermal ectoderm. So the outer layer is called as ectoderm, which is predominantly occupied by the epidermal ectoderm, and a small area in the mid-dorsal line will form the new ectoderm. Then the inner layer. Inner layer consists of the mesoderm, the notochord. So together they can be called as the chorda mesoderm. So the chorda mesoderm, this is the this is the notochord and this is the mesoderm, and the endoderm. The endoderm, it will form the floor and walls of the Archenteron. So this is the Archenteron. So the floor as well as the walls of the Archenteron will be formed by the endoderm, and the chorda mesoderm will form the roof of the Archenteron. So we will just go to a section. So when you take a cross section through the gastrula, this is the Archenteron. This is remember, this is a cross section. You cannot see the blastopore. You take that ball, you just make a cross section. You will be seeing a central cavity, that is the Archenteron. The blastocele is now lost, or it has merged with the Archenteron. So this is the Archenteron. This is the dorsal side of the embryo, where you can see the outer layer is formed by the ectoderm. Majority of the ectoderm is constituted by this epidermal ectoderm, and a small area on the mid-dorsal line is occupied by the new ectoderm. Now the inner layer. So this is the outer layer which consists of the new ectoderm on the mid-dorsal line and the remaining part occupied by the epidermal ectoderm. Now the inner layer. Inner layer consists of the notochord as well as the mesoderm. The notochord on the dorsal side and the mesoderm on the lateral side, and the endoderm. So when you take this as a cavity, the dorsal side of this cavity is occupied by the notochord, or the roof of the cavity is occupied by the notochord. The sides or the lateral walls of the cavity is occupied by the mesoderm, and the floor of the cavity is formed by the endoderm. So Archenteron roofed by notochord. On its lateral walls you can see the mesoderm, here shaded in black, and on the floor and walls you can see the endoderm. You can see this area. This is actually the mesoderm. It has occupied the position between the endoderm and the ectoderm by rolling in. It has smartly rolled in to occupy the position between the ectoderm and the endoderm. Here also you can see that. This has occurred in both directions. Now the endoderm. So this is the endoderm. So this as well as this are the free edges of the endoderm. So this is one free edge of the endoderm. This is the other free edge of the endoderm. The free edges of the endoderm, they will grow towards each other. They will approach each other in the midline. So they grow to this direction, they grow to this direction, fuse, and thereby completely enclosing the midgut. Initially only here you can see the endoderm. These are the free edges of the endoderm. They move towards each other, or they grow towards each other, fuse and completely encircle the midgut. And you have the mesoderm, which is occupying the space between the endoderm and the ectoderm. So this will be like a tube. The outer tube is consisting of this this is like a tube where the outer wall is made up of the ectoderm and the inner wall is made up of notochord, mesoderm and endoderm, and later the endoderm will form the actual lining of this gut, pushing mesoderm between itself and the ectoderm. So, the mesoderm has to come to lie between the ectoderm and the endoderm, because the endoderm also occupies this area. Endoderm will also occupy this area completely encircling the gut, and the mesoderm comes to lie between the ectoderm and the endoderm. So, this is the entire process that depicts gastrulation in frog.